Hello again, and welcome back to the second part of Chapter 4. We're almost there, guys. You will have survived the first 3 chapters that really can knock your socks off in this course. So we're almost there. We're almost to getting into anatomy and physiology full-time. Woo, we can do it. We can do it. So today what we're going to talk about in this part is DNA. Which is odd how I find the book likes to talk about mitosis first before it talks about DNA, which makes me go, okay. So again, we're going to touch on how DNA does its thing. Again, I'm not going to get into too much of the crazy nitty-gritty about it. Like I said, this is kind of like a fly by the seat of your pants refresher on biology, these chapters, which is probably why a lot of people are like, and I get it. But you can do it. I believe in you. So, let's talk about DNA. So, remember, out of the 4 major biomolecules that make up all living things, there's carbohydrates, there's lipids, there's proteins, which we just talked about proteins and enzymes last chapter, and then there is our friend, the nucleic acids, and DNA and RNA. So we're going to talk about DNA. He's the big boy. And DNA, or deoxyribonucleic acid, and yes, I did have a biology teacher who beat how to spell that into me, which is probably, or beat, yeah, how do you spell that? Yeah, because that's, it's a big word, which is why we shorten it to DNA. So, again, notice the nucleic acid in it. That's how you know it's a nucleic acid. Yeah. The deoxyribo means a couple of things. It's actually the sugar, because it has a sugar phosphate backbone, and it's double helix. So that's what the deoxyribonucleic acid is. Now, RNA is just ribonucleic acid, because it's not a double helix. And this is where I also get into my story of how it was discovered, or at least the form. What it did, on the other hand, was something we've been ongoingly working on for quite a while now. But it's interesting because only about 60 years ago, we discovered how it's put together in that beautiful double helix that we know and love today. Like my little plushy guy right here. Anyway, so, it's held together by complementary base pairs, which are known as the purines and the pyrimidines. So, I like to call them the biggies and the smallies. So, what's going on? Or I call them the fatties and the skinnies, really. Anyway, so these guys always match up in certain ways. And a purine always matches with a pyrimidine. And now you'll be noticing, wait, is there 4 bases? Yeah, there's 4 bases. Then why are there 3 pyrimidines here? That's because this guy right here, uracil, you don't meet him until we talk about RNA. He's just kind of like the weirdo. Again, remember I say in science, or biology, it's always and but. Yeah, our uracil's your butt right here. So anyway, in DNA, adenine always goes together with thymine, and guanine always goes together with cytosine. So A, T, G, C. And the reason we don't have like 2 purines going with each other is because it would actually make bulges in the DNA, and that's not good for reading the DNA, because we're going to talk about in a minute how we open it up and enzymes go in and read it, and also make duplications and whatnot. And the last thing you need is like bumps on the road when you're already reading it and you're going, it's not that good. So that's why A always goes with T, and vice versa, and G always goes with C, and vice versa. A purine always goes with a pyrimidine. So we don't get bumpy, lumpy DNA, which would make life so much harder on the enzymes. So how does DNA replicate? Well, this was an interesting story. Basically, we had to, this is where the adding something to the DNA to make it kind of glow was one of the reasons we kind of figured it out. There's a lot of very classic experiments that figured these things out. That, like I said, in biology, I'd go deeper into, but this is anatomy and physiology, and I just need you to know this so we can move on and talk about that. So anyway, So basically, every time, as we were talking about mitosis, every time the cell splits, there's a whole time where the cell has to duplicate all of the DNA, make a copy of it. Well, how does it do that? It uses itself as a template for the copy. And that's called semi-conservative replication. So what's happening here is it unzips, and that's what we literally call it. There is an enzyme called helicase. Guys, remember, if it ends in ase, it means it's an enzyme, like polymerase. And you'll see that over and over again. If it ends in ase, it's a tip-off that that is an enzyme. I just thought of the thing I couldn't think of in the last lecture. All right, sorry. That's how my brain works. So anyway, so helicase unzips it. And then a guy by the name of DNA polymerase comes by and attaches, and he starts adding free nucleotides, and we actually get these from eating food, by the way. Yeah, we actually get DNA by breaking down other what we eat's DNA and recycling it into our own DNA. So if you're wondering where all these base pairs come from, we ate them, and then we turn them into our own DNA. Yum. So anyway, so it's called semi-conservative replication because, like I said, we build the new strand based off the one side of the old strand, and the same thing's happening on the other end. And what's happening is, unfortunately, DNA polymerase is really good at going in one direction. And that one direction is called the leading strand. He's really, really, really good at going from 5' to 3', because there's we have parts that stick off the DNA. Like if you go back here, like right here, if it's pointing that way, it's called 5'. If it's pointing that way, it's called 3, or the opposite right here, this OH sticking out, that's called the 3'. It's named after like how you count the sugars. Like it's like 1, 2, 3, and this one would be 1, 2, 3, 4, 5. So it's how you count that sugar right there. So that's where that 5' and 3' come from. And the polymerase is really good at going in one direction, but he's like me trying to drive a stick shift in the other direction. So what happens is we get a replication bubble, and we've got one polymerase going this way, another polymerase going this way, and so they're doing it at the same time. And this actually happens in several spots. This isn't just one strand of DNA. It's actually one strand with several of these guys doing these bubbles at several places until they completely open up, and the brand new strand pops off the other strand that's made of new and old. I hope that makes sense. So again, this part, like I said, imagine there's more than one replication bubble happening, and they're all happening down the chain, and eventually all the polymerases meet each other. It's kind of like two trains going down. One's going that way, one's going that way, and it keeps zipping, and it's just building two new strands using the old strand as a template for the other half. I'm hoping this is making sense. Again, I have videos to recommend for this because it helps. Anyway, now, like I was saying here, the leading strand, like I said, DNA polymerase is going in one direction really well. He's really good, and that's called the leading strand. Unfortunately, he sucks at going backwards. This is just one of those things in nature where I like to point out that it's inelegant design. This was just kind of like, well, you can do it. And polymerase is like, yeah, I can, but I don't like going in that direction. I don't work very well going in that direction. And the body's just like, well, we're not making a new one of you, so go. So it's kind of like me driving a stick shift. It's like, because, I suck at that. And I almost drove my dad's truck into a ditch because I suck at it. So anyway, so yeah, it's going this way, but it's doing it like stop, go, because he can't do it very well in this direction. He sucks at going in this direction. And because of that, we make these things called Ozaki fragments, which is named after a couple of scientists. The husband's from Japan and the wife is, I forget where she was from, but she's not Japanese. But the two of them basically figured out this part, the lagging strand, and why polymerase goes terrible backwards. And and then another enzyme comes along and actually fixes all these fragments back into a proper strand. So polymerase goes really easy one way and terrible in reverse. So, and then you get the Ozaki fragments. So that's where we get the leading strand that goes fast and the lagging strand that is like somebody trying to drive a stick shift that is clearly never driven a stick shift. But eventually we get there and we make a whole 2 new strands of DNA with both old and new inside of it, and that's called semi-conservative replication. And hopefully that made sense. Like I said, here's the steps. Am I going to beat you with these? Not so much, but be aware of helicase. He's the one that unwinds. DNA polymerase, he's the one that puts it down in the 3' to 5' direction. And then because he's terrible at going from 5' to 3', he has to have a friend that helps him along, which is ligase, who stitches together, again, ends in ace. He's an enzyme, and he stitches together the Ozaki fragments, as I was talking about. So again, if you're confused, and hopefully I didn't confuse you, I'm sorry if I did. But definitely watch this video on DNA structure and replication. He has a really good one on it with animation, so you can actually see it happening in real time, which is really nice, and funny sound effects. Highly recommend. Highly, highly, highly. So that way, if you need a visual animation with this, and I highly recommend you do, watch this one. All right, with that said, how do we use DNA? As I mentioned earlier, you kind of saw some of that in the video I showed you from last chapter, where I got really super giggly over the transport enzymes. So what's going on is that DNA is like a cookbook. And that's why DNA is so carefully hidden away in us eukaryotes in the nucleus. Because we don't want them to lose them. We don't want him to leave. Because he's filled with all the things, all the instructions to make all the proteins that make us up. And as I was showing you in the videos, we have a lot of proteins. We have enzymes that are constantly doing jobs. We've got, a lot of proteins sticking out of the cells, allowing the cells to do a whole bunch of different things. So we're always talking about enzymes. We're always talking about proteins. And where all of these guys come from is from DNA. So there's basically kind of 3 steps going on here. And it's just like cooking, sort of. And that is this. First of all, we need the cookbook. DNA is the cookbook. But imagine it kind of like grandma's cookbook. The one you don't want to lose because it's been passed down through the family, or it's a really good cookbook you really don't want to take out of the house. And imagine your house is the nucleus, and your cookbook is the DNA. So, what do we do when we need ingredients out of the recipe in the cookbook? Well, you make a list, right? You don't bring the cookbook to Ingalls. You don't bring the cookbook to Harris Teeter. No. You've got to make a list. That list is the mRNA. So there's 3 flavors of RNA. Actually, there's 11, but we don't care about the other ones. We only care about these 3. So, yeah, I found out there's 11. Actually, there might be more, but we care about these 3. These are the only 3 we care about. So anyway, so there's messenger RNA, who's the one that goes in and actually the DNA unzips, again, helicase comes back, and he makes a pattern of himself off of the DNA, and then that DNA then goes ahead and goes out, the mRNA goes out after some editing and finds a ribosome. And this is what I pointed out in that video in the last chapter. Is that the mRNA that we saw coming out of the nuclear pores was the mRNA, and the mRNA went and he found 2 things to float around and attach. That was the small and the large subunit of a ribosome, and then it went around the loop to read it, and as it did, it was making a protein. So the ribosomal RNA is inside the ribosome, by the way, and that's why it's called ribosomal RNA, and that's kind of what matches itself to the mRNA that just came out of the nucleus from the DNA. And then these things that look like T's are called transport RNAs, and they actually bring in an amino acid, because remember what I said about back in chapter 2, if you recall, because I've been filling your brain with lots and lots and lots, and that is this. The transport RNA brings in an amino acid. And that's what we saw coming out of the ribosome, is these guys come in with an amino acid attached, and they have a code that is always 3 base pairs, and that is a codon, and the anticodon's at the bottom. It attaches and matches, and that's literally codes for one amino acid. And the amino acid attaches into a chain, and then it releases and turns and folds into a protein, as you saw in that video in the last chapter. So that's called transcription and translation. So transcription is basically inside the nucleus. It's where the DNA unzips with helicase. Then an enzyme comes through and makes a copy using mRNA, messenger RNA. So it makes a copy of this one portion, and there's a whole bunch that goes into this. How do you know what part? La, la, la. There's a whole bunch of it, but we're not going to get into that. But just know that mRNA comes in and kind of reads the DNA and patterns itself after the DNA, and then unhooks, and then it usually gets edited a little bit. Again, we're not going to get into that bit. And then it leaves the nucleus through the nuclear pore. And then it goes and finds a ribosome, and that's what begins translation. The ribosome comes over, hooks on, and starts reading it. And as he's reading it, he's calling out for amino acids that every 3 base pairs code for. He's like, I need, this, I need this. And the transport TNAs bring those amino acids in that match what he's calling out, and then he puts them all together while he's reading the mRNA into a protein strand that eventually leaves. And then we saw that in the video from last chapter. That's what I pointed out. That's called translation. So transcription, inside the nucleus. mRNA is made off of the DNA. Translation, the mRNA comes out, finds a ribosome, and through that, and the help of tRNA, makes a protein. And remember, several ribosomes can actually read the same string. They only showed one at a time, but several ribosomes usually go crazy on it. So again, a good example of something that can definitely is translation, transcription. Sometimes, again, videos help immensely with animations because I can't animate with just my voice alone and my face. So, however, there is a couple things I want to get into that before I move on, and that you'll see on the test. So let me go back to the Jamboard from last time and erase it. How do I erase? There's got to be a faster erase. Clear frame. Duh. One of these days. So anyway, there's something I want to do, and that is, how do you do translation and transcription with DNA and RNA? Because there's going to be a couple of questions on your test on this. And this is kind of hopefully a blast from the past. So remember, DNA, and if it's not, I can go more into this a little bit. So remember what I said. In DNA, A, adenine, always goes with thymine and vice versa. And G, guanine, always goes with cytosine and vice versa. So, for instance, again, we read DNA in sets of 3. It's called a codon. And the opposite of that is called an anticodon. So if, for instance, I say, and you'll see this in a test question, give me the complementary base pairs of a strand of DNA. That just means I need the opposite strand of what I give you in that question. So for instance, if I just go, let's see, ATG, CCA, A, GTA, oh, I need to make this longer. Okay, so if I give you this, and I want the opposite of that, that's basically when I say a complementary base pair. So if I say, please find the complementary base pair of what I just gave you in a test question, I'm asking for this. I'm asking for the opposite. So for instance, let me move this down. So you just use this right here. And that is, so if I see an A, I'm going to put its opposite, which is the T. So literally when I do this, I put T, and there you go. That's the opposite. When I see a T, I'm going to put an A, because again, that's the opposite. When I see a G, I put a C. This is starting, hopefully, to make go, oh, okay, yeah, that makes sense. Same thing here. So if you want to, go along with me, if I go, so the opposite of C is G, so G, G, the opposite of A is T, again, C, A, T, and G, T, A, C, and yeah, there you go. We just did the complementary base pair right here of the one on top. Yay, you got it. Now, this is what happens when we're replicating DNA. Okay, so that's why complementary is a tip-off. We're talking about DNA replication. So this is what's happening when the DNA opens in a bubble and we're making a replication during cell division, fun stuff like that. Now, let's go ahead and talk about a different crazy person and when we're doing translation and transcription. Now, remember, we're dealing with somebody else because RNA doesn't have thymine. He doesn't have T. He has U. So A equals U, uracil, that guy I mentioned earlier that I said we'll talk about again when we talk about RNA. We're here now. So RNA, let me make sure my face isn't in the way of this so I didn't put it. Oh, good. It's just under it. I didn't want to put, start typing up where you can't see it. That would be silly. Anyway, so again, if I give you this and I say, for the DNA line, give me an RNA line. So again, if I just give you a DNA line, so I'm just going to go ATG, CCA, ATG, right, move it, ATT. Boy, I'm sorry, I'm kind of in the There we go. There we go. No, that's not it. Why did I put a Y? All right, so there you go. So I just gave you, a DNA. And I say, hey, what is the mRNA going to look like off of this DNA? And you're probably going, well, even though the DNA still has T's, RNA won't have those T's. So when you see A, because RNA doesn't have that, he just goes to what he does have, and that's U. So when you see an A, you're going to put down, and you're doing RNA, you're going to put down a U. Match. There we go. But when you see this T right here, you go, but yeah, we do have the opposite of T in RNA. It's just A, so we put A again. And G and C are still best buddies forever, so you don't have to think about that hard with them. So again, it's GJ. Now again, you look at A, there's no T, so you got to put down what we've got instead, which is U. So again, U, and again, when you see T, you're still going to put down A because we still have A, G, C, and then again, A. All right. Hopefully that's clear. Is that a bit clear? So remember, when you see an A, you're going to put down a U if it's RNA. So if you're going from DNA, RNA, that's the only difference. And you should have no T's on this opposite end. If you've got some T's and you're supposed to be doing RNA in the line you just made, then you're not doing it right. RNA doesn't have T. He only has U. Now, if you're going to go ahead and there's a third type of question you can get on the exam, and that is RNA to RNA. Again, we just ignore the DNA that's not there anymore, and again, we just be like, UAC, UUG, CAA, stuff like that. Again, you're going to be going RNA to RNA, so it's not a big deal. So again, there should be no T's whatsoever. So again, you see a U, you put an A. You see an A, you put a U. G, A, C, G, U, U. Goo. That was weird. Anyway, I think the skeletons are getting antsy on me. Anyway, so there you go. That's the difference between the two. So that way, hopefully I've explained it well enough that you can kind of see, because this will pop up on the test. You will see this on the exam. So remember, complementary, if you see the word complementary, it just means going DNA to DNA. If you see anything talking about DNA to mRNA, that's what this is. And that's where you have to be mindful of the A's, the T's, and the U's. And then if you see from mRNA to the enzyme of ribosome, that's just RNA to RNA, okay? I wanted to show this to you because, yes, this will be popping up. And I'm probably going to put in some of the questions from this lecture, a couple of these for you to do yourself. So that way you're not going, I have no practice. So I'll give you a little bit of practice on that. All right, with that said, where was I? Right. So we were on, what happens if we screw this all up? Well, that happens. And interestingly enough, our body actually has several enzymes that actually double check the progress of polymerase. So we have checkers. So it's actually kind of funny because DNA polymerase actually checks itself. It's actually so huge, it has a built-in before and after checker. So it's not only pre-checking, it's after checking as well. And even then, we get something when sometimes the base pairs get mixed up. Sometimes it doesn't mean a lot. Sometimes it does. And this is where mutations come in. Mutations can both be good and bad. Mutations occur in one of two ways, spontaneous or induced. Spontaneous is just where, like for instance, you've been sunbathing and unfortunately the sunlight does have enough energy to punch DNA. And when it punches DNA, it punches out parts and then an enzyme has to go through and actually literally put things in. We do this a lot. We actually slap a lot of different things in. It's actually really interesting. Again, I could go into this quite a bit, but you just know that, just living, unfortunately, brings us to spontaneous mutations. It happens all the time. Your body actually has ways to deal with this. So it gets usually fixed really fast. It isn't, unfortunately, until you're older when your body slows down is when you're not dealing with those as well, which can lead to cancer. Induced, on the other hand, can be caused by mutagens, which is like chemicals, radiation, falling into a giant chemical vat and turning into the Joker. No, that's not it. Anyway, like I said, simple mutations get fixed through DNA repair all the time. We're constantly doing it. It's happening all the time. We're fixing it all the time. But some mutations can never be fixed, and especially if they're passed on to your kids. So there's a lot of different things that can happen. You can have deletion where an entire section, like these are all chromosomes, by the way. So like deletion can happen, an entire section of a chromosome is just gone. Duplication, it's kind of like in reverse. So it just, for whatever reason, they copied it twice. Inversion is where one, section gets flipped. That happens a lot, but that also may not be a bad thing. Some of these are bad. Some of these are actually allow for genetic variety to make us all different from each other. So that's a good thing. So we'll get into when it's good and when it's bad a little later. But just know that not all mutations are going to lead to cancer and then you drop dead or give you cool superpowers. Unfortunately, no mutations have given us cool superpowers. Except for the ones that make us live. But anyway, living is a pretty cool superpower. Insertion, like for instance, here's chromosome 4 and chromosome 20, and all of a sudden part of chromosome 4 just got lobbed into chromosome 20. And then there's translocation, where they just literally switch ends. This is not a bad one. Insertion is interesting. Deletion, duplication. Duplication's not bad because usually it ignores it. Deletion's pretty bad though. So like I said, some of these things can be really, really good for us in the long run, especially talking about genetics, and some things can lead to cancer, and that's really, really bad. So mutations is kind of like a good, bad thing. It's good because it leads to increased diversity, genetically speaking. It's bad because in the short term, some of it can cause cancer. So yeah, induced usually is something our body doesn't deal with very well. Spontaneous, we have literally redundant systems to set up and fix this. Induced, if there's enough damage, Yeah, it's not good. Not good. Not good, Monty. Not good. So anyway, again, he explains this, the different types right here, and the Bozeman science. Good video, good video. He likens it to like a cookie recipe. So if you really like that analogy, if it's kind of like a cookie recipe, I highly, again, recommend watching this. But I'm not going to go crazy with the different types of mutations. I might, give you a couple of questions of like, what is a mutagen? Although I haven't looked at the test in a minute, so I'd have to go back and look. But anyway, other than that, there we go. We've just survived Chapter 3 and Chapter 4. Congratulations, you did it. Rock on. Oh, Monty yawned. Monty yawned. There's a hair in your mouth. Can I get that? Thank you. With that said, thank you. And I'll see you next week when we talk about Chapter 5, which is basically how cells stick together and stuff like that. And then Chapter 6, where we start with the integumentary system. Woo! Which is our skin. So with that said, I already said goodbye, but I'll say it again. See ya. Bye.